Hello everyone and welcome to another class. In this class we are going to learn about tobacco mosaic virus. Okay. This tobacco mosaic virus is very important virus and it is one of the most researched virus in virology. Okay. And this virus contain 1.63 proteins per turn and it is a rod shaped helical virus. See, rod shape means it is a cylindrical virus and it has 16.3 proteins per turn means if we say this is one turn, okay, so the proteins will be arranged like this, okay, such that it contains 16.3 16th and a part of 17th, you can say, proteins per turn. Okay, so basically I first will show you how this affect the leaf. Okay, let's wait. So right here, as you can see, this is a leaf of a tobacco plant and it shows some discoloration here. This is what happens if this virus infect tobacco plant. Okay, and this virus contain a single stranded RNA genome with a 7300 nucleotides. See, this is the RNA here. It is a single stranded RNA. Okay, usually the genome contain double stranded RNA. This is the one strand and this is second strand. Okay, but this tobacco mosaic contain only one strand, single stranded, and this one right here is double stranded. So this is not the case, and here the case is of single strand, and this is a positive sense RNA. Okay, and it contains 7300 nucleotide means if you count it like this, like we have first A, then U, 3, 4, 5, 6, and if you continue like this, it contains 7300 nucleotides in it. Okay, now the transmission here is, first of all, it transmits through. Transmission means how it spread from one plant to another. The first point is contaminated equipments. If the equipments which the farmers or field workers are using, they are contaminated from this virus, then this virus can spread from one plant to another. And the second point is workers hand, right? Not all time they need equipment, so sometimes they use their hands also, and in that case hands can also spread this virus. Third point is leaf rubbing, and this leaf rubbing is very, you can say, natural. Sometimes wind flows and if two plants are in close proximity to each other, their leaves can rub against each other. So if this plant is diseased, then it can transmit its disease to a healthy plant also. And the third is wounded plant and infected plant contact. Okay, somehow a wounded plant, let us say a farmer is farming tobacco, right? And if that farmer cuts the diseased plant and put it somewhere, or let's say just cut it and place it up in a part of his or her field, right? So in that case, if that wounded plant comes in contact with the plant, healthy plant, then this can also spread the disease, right? And that wound can be on both plant also or in any of the plant. Okay, now what happens inside the host is, when virus somehow through these transmission methods comes in contact with the new plant, it enters that tobacco. Okay, and after its entry, the capsid will dissociate. So this outer part is capsid. Okay, this will dissociate and just get destroyed, and this lead to the release of this mRNA, not mRNA but RNA. It's nucleic acid into the host, which is tobacco, and viral RNA behaves as mRNA since it was a positive strand. Okay, so it behaves as mRNA. Thus, ribosomes gets attached to it, and see, ribosomes has two units, large and small. They get attached to this RNA. Okay, and they form proteins. The first protein formed is the replicase protein. Okay, now this replicase protein, it attached to the 3' end of RNA. Okay, this RNA has two ends, right? This is 5' end and this is 3' end. So usually this ribosomes will attach from 5' end, but if later some proteins are found, right, replicase, it will attach to here, replicase. Okay, so and this will then kind of replicate it, and from this positive strand it will make a negative strand RNA. Now this negative strand RNA act as template to form positive sense. Okay, so here is my negative sense RNA. Now when it replicates, it will always form negative sense RNA. this was positive, and from positive it will form, oh sorry, this was negative, it will form positive sense which can act as nucleic acid also, right? And this replicase can also help in formation of its coat also, right? After this positive sense translate, it can form, okay, this positive right here, this positive sense can translate again to form two kinds of protein. The first is coat protein and the second is movement protein. This coat protein helps in viral assembly, right? So here is my nucleic acid and here is my DNA. So what this coat protein will do, it will merge them and form a full-fledged virus. And there is membrane protein which integrate with this full-fledged virus, and what it will do is it will again it will again help this virus to infect the new host or the cell of that same host, okay? So here is what I have drawn, that mRNA will enter the cell, the negative strand RNA, not mRNA, but it's simple RNA. Okay, this is virus here. It will enter, capsid will degrade, RNA will release, ribosomes will attach to it, and first protein which is formed, which is that is replicase protein, and this will help it to form positive sense RNA which can act as template. Then it will form coat protein, movement protein, and also negative sense, many negative sense RNAs, right? So many negative sense RNAs, since they are formed, it will attach with the capsid to form virus, okay? And this movement protein will help in exit of this virus from here, and can also help in affecting new host, affect new host slash cell, okay? Because one plant contains many cells, so if it attacks on a cell, like right here I'm showing, like let us say it's attacking this cell, so it can attack next cell also in the same plant. So that's it for this video. Thank you very much.